Hi, I'm Don Clavin, the Town of Hempstead Supervisor, and welcome to the Town of Hempstead's Shellfish Hatchery. We're proud of our efforts here in the Town of Hempstead to clean our waterways. So we put together a series of videos for you to sit back and enjoy and see exactly what we're doing.
You know, I grew up about a mile or two from here. I grew up on the water. I'm very fortunate, very lucky now that I get to work doing something that actually protects and benefits the bay, benefits the marine resources that a lot of other people in our township, love and share. Shellfish are part of our maritime heritage. People have always come down to the shore, to the bay, to feed their families, feed themselves, make a living. And that tradition even continues today with our modern clammers, our recreational and commercial clam diggers. The town issues about 1,500 shellfish harvest licenses each year. It's a big pastime recreationally for people, and there's still quite a few guys out there that make a living digging for clams every day.
Why are clams so important to the town's waterways? Clams play a crucial role in keeping our waters clean. These bivalves are effective natural filters, capable of removing particles as small as 3 microns from the water column. Each clam can filter up to 25 gallons of water per day. But that's not all. Clams also help remove excess carbon and nitrogen from the water, incorporating it into their shells and tissues. As they filter feed, they consume tiny organisms and organic matter, which are broken down and excreted as waste. This process helps to improve the quality of the water and reduces harmful algal blooms and oxygen depletion. So how do they do it? Clams use their siphons to pump water through their gills, where tiny hair-like structures called cilia trap particles and move them towards the clam's mouth. The clean, filtered water is then expelled through another siphon. This process not only cleans the water, but also provides a habitat for other marine organisms by creating a stable and oxygen-rich environment. So the next time you're enjoying some fresh clams, remember that they're not just a tasty treat, but also important for the health of our waterways. January marks the beginning of the Town of Hempstead's hatchery operation at the Department of Conservation and Waterways. Adult clams are collected from the wild in Hempstead Bay. These clams are then brought into the hatchery facility, where they will eventually become the parents of the millions of baby clams to be spawned during this process. It's important that we spawn local clams from Hempstead Bay because these clams are already adapted to the unique environmental conditions of our waterways and have survived here for generations. After the clams are harvested, they will be moved into a conditioning tank for 6 to 8 weeks, where they will feed on a mixture of 4 different species of microalgae created in the hatchery's Algae Culture Lab. Algal culture is probably the most time-consuming and challenging element of the hatchery operation, but it is absolutely essential to its success. The Shellfish Hatchery's algae cultures will become the food source for the adult clam broodstock. These cultures are grown within a continuous algae production system consisting of 20 200-liter bags, which are assembled, sterilized, and inoculated by hatchery staff. The algae are very colorful and grown in suspension, with air and CO2 bubbled into each bag for proper gas exchange. Once a week, hatchery staff take samples from each individual algae culture bag in order to measure the concentration of algae cells in each bag. Counting cells is an essential technique in many scientific environments, helping to achieve accurate, reproducible, and statistically relevant data. The most common way to count cells is by using a hemocytometer, an instrument that bears two laser-etched grids which aid in the counting of a small sample of cells under a simple light microscope. This data can then be used to extrapolate the number of cells in the larger experimental sample. After putting a known volume of water onto a slide, samples are then examined under a microscope, where staff do a hand count of all visible algal cells using the hemocytometer's grid system as a guide. This is followed by a mathematical extrapolation, which determines the larger cell count by using the information gathered from the smaller sample. This process helps determine how much algae to feed each tank per day per clam. In addition to cell counts, algae cultures can be assessed visually. Each bag lasts approximately 3 months before they crash. Changes in color often indicate a loss of density or contamination. For example, a bag that starts a dark golden brown will end up light orange by the time it crashes. The hatchery's inoculation process involves the cultivation of algae strains under optimal growth conditions in order to provide all the nutrition needed to feed our broodstock and juvenile clams. The process begins with a small flask that contains a sterilized mixture called F2 media, which is comprised of seawater mixed with micronutrients. Next, a small volume of a homogeneous culture acquired from a federal microalgae collection is added to the F2 media. Our clam hatchery cultures up to 6 different species of microalgae. The algae will become more dense and concentrated over the next 7 to 10 days as it metabolizes the micronutrients inside the flask. This new mixture is then transferred from the small flask to an intermediate flask, from there to a larger carboy container, and finally from the carboy to the larger volume bags in the algae room. Each of the Algae Culture Lab's 200-liter bags produces a runoff of algae that travels down a series of small gutters. It then proceeds to a larger gutter on the wall, where the food supply is eventually collected in a harvest bucket. When the level of algae collected reaches a certain threshold, the contents of these harvest buckets can be pumped into any location in the hatchery facility. After an abundant amount of the algae food source has been prepared, the adult broodstock take up residence in the hatchery's conditioning tank for approximately 6 weeks. Conditioning is the process of inducing gametogenesis, which is the ripening of gonads into gametes in the form of sperm and egg. While feasting on the algae, the clams are also building up their energy reserves, and when they are deemed to be fully ripened, the clams are then moved to the spawning tank. By manipulating the water temperature in the spawning tank to simulate the warmer summer conditions in Hempstead Bay, the hatchery staff trigger the shellfish to spawn or release their gametes. Once they begin to spawn in the large tank, clams are then removed to individual beakers so that the hatchery staff can control fertilization. The hatchery staff meticulously rear the developing animals throughout their entire life cycle, from the moment the eggs are fertilized to the various stages of the larval cycle and metamorphosis, up until the juvenile clams reach seeding size. The larval cycle consists of 4 distinct stages, beginning with the trochophore stage. After fertilization, cell division occurs rapidly, and within 12 hours, the embryos develop into free-swimming trochophores. During this stage, the larvae are covered with cilia for movement, rely on a yolk sac for nutrition, and their shells are not fully developed yet. The early trochophore stage is ideal for dispersal, as lingering around the broodstock too long could make them vulnerable to consumption by larger filter-feeding animals. Within 2 days, trochophores grow into the next stage, known as straight-hinge veligers. The animal is now protected by a thin, transparent shell that covers its organs. The shell shape resembles a capital letter D, and a cilia-covered organ called a velum has developed. This vellum serves both for locomotion and feeding, as it captures food particles and propels the animal through the water. The umbovillager stage follows next, marked by a changing shell shape as more and more layers are built up. The hinge area, known as the umbo, becomes more pronounced and robust. Larvae in the hatchery typically reach the umbovillager stage nearly 4 days after fertilization. The larvae continue to grow rapidly, almost doubling in size every 2 to 3 days. The final stage before metamorphosis is the pedivillager stage. During this time, the animals can both swim and crawl. They have developed a foot used for crawling along the bottom, sensing their surroundings, and finding a suitable spot to burrow. Meanwhile, the vellum begins to shed, and the pedivillager relies more on its new gills for feeding. Overall, this life stage is adapted for leaving the water column and transitioning to the benthic habitat. Throughout the entirety of the larval cycle, the hatchery staff takes great care to provide proper nutrition and maintain a clean and favorable environment for all the animals to thrive. Each day, the tanks containing larvae are drained, cleaned, and fed. Mesh trays with different size screens are used to sort and separate the larval population by size, redistributing them back into the conical tanks at suitable densities. This prevents smaller larvae from becoming outcompeted for food by larger ones. The health of the larvae is also closely inspected, and any sick, dead, or stunted animals are removed from the population. This culling process favors the growth of healthier, faster-growing animals while removing the unhealthy ones. Ultimately, only the strong survive and progress to the next stage, metamorphosis. Metamorphosis is a significant developmental milestone in the life cycle of the clams, signifying major physical and physiological changes for the animals. Sense organs, musculature, heart, kidneys, nervous system, and digestive tract all reach peak development. Once metamorphosis occurs, the hatchery staff move the animals from the larval conical tanks to the floating downweller trays, allowing them to spread out, grow, and feed more efficiently. These static tanks are filled with water once a day and fed with algae from the hatchery's Algae Room. While the clam's growth continues in this downweller environment, it is merely an intermediary stage and certainly not the most efficient way for the clams to grow. After spending about a month in the downweller system, the clams will reach approximately 1 millimeter in size, at which point they are transferred to the more efficient upweller system. The upweller system is a multi-part process involving both indoor and outdoor components, and is a much more efficient way to feed the clams, ensuring that they also grow at a faster rate. Unlike in the downweller, seawater is continuously pumped to the indoor upweller system from the bay 24/7 through pipes that then deliver the oxygenated water full of naturally growing or ambient algae to the system. When the clams outgrow the indoor tanks, they are then transferred to the silo-based outdoor upwelling system. There are mesh screen bottoms of various sizes on each silo. The clams are sorted and separated by size to prevent competition for feeding and survival. A recent count of the upweller silos combined for a total of over 2 million clams. The clams will be resorted by size at least once a week, and they will enjoy a constant flow of nourishing algae-rich water that keeps them feeding around the clock in a kind of always hungry mode. As a result, the clams grow at a very fast rate, which then necessitates that they be moved again, this time to a floating upweller system, or Flopsy as it is known at the hatchery facility.
This Flopsy in particular was built in 2008. It was designed by the previous hatchery manager. It's one of the largest Flopsies in Long Island. This is a much larger version of the clam and shellfish nursery that you saw in our hatchery facility. This actually floats in the water. It moves a much larger volume of water through the growing clams, and it provides them more food, more algae, oxygenated water. It keeps the clams feeding 24/7. They're constantly feeding in here. It's a very effective way for growing clams, and it keeps them protected from predators. So these clams will continue to grow in our Flopsy throughout the summer and fall.
This past July, the hatchery reseeded 2 million clams into Hempstead Bay, completing the vast circle of life in our town's local ecosystem.
So this is the end of our season, and we've spent the last 8 months growing these clams out in our nursery. They've been protected from predators. Every day we've been washing them down and sorting them by size and giving them just the right conditions that they need to survive. And we've had very low mortality as a result. We've given them optimal growing conditions. And today's the day we're going to start to put them out in the wild, back into the clam beds. It's very satisfying to watch them grow. You know, you've seen at the beginning of this process, hatching from eggs, and now they're almost about a quarter in size. You'll see we've got some nice clams growing, and I'm really excited to return them to the bay. Over the last 30, 40 years, this program, it's expanded and it's been widely successful. Today, we now have one of the most productive clam beds in all of Long Island. And we have to thank not only our staff, but all the administrations that have continued to sustain this program. Each year, we put more and more clams in the bay, and it's been a benefit to all our residents and to our water quality. And over the next few years, we'd like to continue expanding our program and ensure that it continues long into the future, so future generations can continue to enjoy the town's marine resources like they do today and as they've done in the past.